The Lac Operon is one of the most tested examples of prokaryotic gene expression on the AP Bio exam. And in this video, we're going to be breaking down exactly how E. coli decide when to turn the Lac genes on and off. Hi, I'm Miss Parrott, and today we're taking a deep dive into the function of the Lac Operon. The Lac Operon is how bacteria regulate lactose metabolism, right? And lactose is the sugar in milk and milk products. So here's a quick example, a diagram of what the Lac Operon looks like. I did a whole video on the structure of Lac Operon, so please do check that. Starts with the cap site. We are going to be looking deeper later on in this video at the mystery of the cap site, but basically it just helps RNA polymerase bind. Then where is it binding to? The promoter. That's the non-coding region where RNA polymerase will bind. Then there's the operator where the repressor binds. And then you have your three structural genes that code for the three Lac proteins that help in lactose metabolism. So a reminder, what's the point of an operon? It's to help the cell use their energy efficiently. So enzyme production, the production of those lactose proteins takes energy. So it's like it takes energy to make energy. And lactose is not always present, right? Lactose is the two 6-carbon molecule that's made of galactose and glucose. So when we break it in half, glucose is now available. That's why we break lactose down in the first place. So, but lactose isn't always present. And glucose really is the preferred energy source for E. coli. Now, there's two environmental signals that regulate when this operon, the operon is on or off. So if lactose is present or absent, and remember, so the environment is not like the weather. The environment of these bacteria is inside of your guts. So is lactose there or not? That will impact its on and offness. And then is glucose present or absent? That will also impact how high or low it's turned on. Okay, so first, the Lac Operon's default state is off, okay? And so, because it's assuming that we're not always having lactose. So, in order to be off, the repressor protein has to be attached to the operator. And when that is there, it's blocking RNA polymerase. Like RNA polymerase physically cannot go past the repressor protein. So this is what it's going to look like if it's off, right? The repressor protein is present, RNA polymerase is blocked. So it cannot transcribe the structural genes. And if it can't transcribe the structural genes, lactose cannot be broken down. This is called negative regulation. This is when we have a repressor blocking, and without lactose, you're going to get no gene expression. Remember, another word for gene expression is protein synthesis. So you're not going to get the proteins that these genes code for, they're not going to get made. This is the default. But when I drink some milk, or eat a grilled cheese sandwich, or have some ice cream, and now lactose is present, this can turn the operon on. Now, how does that happen? Lactose will bind to the repressor protein that's here on the operator and change the repressor protein's shape. If the repressor protein isn't shaped to where it can jump onto the operator, it has to come off. So when lactose is present, it changes the shape of the repressor and they come away. Now, RNA polymerase is no longer blocked and it's free to move along the DNA strand and transcribe the lacZ, lacY, and lacA proteins or genes into mRNA, and then the mRNA will get transcribed. And usually it'll go back and it'll just keep going as long as it's needed. So this is called an inducible operon. Inducible means like turn onable. Think of it like a light switch. The default is off, but when you need to go in the room, the light switch turns on. So the RNA polymerase is there, and now we're going to transcribe. So RNA polymerase, right, binding to the promoter, going past the operator. Then we have, again, our lacZ, lacY, and lacA genes. Those genes are transcribed, right? Transcription is when we go from DNA to, in this case, one long messenger RNA. Again, that's part of the efficiency of using operons and why bacteria have them. So we've got one long mRNA, but we have 3 different ribosome binding sites. So ribosomes will bind at the beginning of each of these, and then it will translate, right? Translation is when we go from mRNA to a protein, and it will translate into the 3 different proteins. You don't have to memorize the names of the proteins, but it is good to know that, like, the first one, lacZ, codes for the production of the actual, the actual enzyme that cuts lactose into glucose and galactose, because the whole point is to free up glucose so that the cell can use it for energy. Then we have lactose permease that goes and embeds itself in the cell membrane so that more lactose can come in. And then the last one is called transacetylase, and its job is to detoxify the byproducts of lactose metabolism. All right, as promised, this is where the capsite and the glucose comes in. So, reminder, we said that glucose is the preferred energy source. If glucose is low in a cell, so it has low available energy, because of a biomechanical reason that could be its whole own video that we're not getting into today and is beyond the scope of the AP exam, we have high cAMP, which is cyclic AMP, cyclic adenosine monophosphate. So this is actually a derivative of ATP, adenosine triphosphate, but instead of 3 phosphates, it only has 1. So if we have low glucose, so our E. coli, they have low glucose, they're going to have a lot of cyclic AMP, right? That's a relationship. Now, there's a protein called the cap protein, and when cyclic AMP attaches to it, the cap protein becomes active and will activate transcription of the Lac Operon. So the strongest expression of the Lac Operon is going to happen when glucose is low and when lactose is high. Remember, this cap protein camp teamwork attaches to the cap site and acts like a flag. Hey, hey, RNA polymerase over here, come. And so then it will come and bind at the promoter. So this ensures that E. coli are using up all its glucose first for energy, and not wasting any energy, again, doing the whole Lac Operon situation unless it actually needs to. So this is it's both a flag to get RNA polymerase to come over, but it's also, this system helps us not use lactose for energy if glucose is plentiful. So that's really smart. So to wrap up and just give like a visual summary, the environmental signals that control gene expression in E. coli for the Lac Operon are lactose and glucose. So if lactose is absent, so we haven't drunk any milk, there's no lactose around, and we have any glucose, the operon will be off. If lactose is present and glucose is also present, remember, if we have glucose, then we're not going to make very much cyclic AMP, so our cap site's not going to be waving the flag. We'll have low. So RNA polymerase will still be able to bind to the promoter. The repressor will be off, but you won't have that extra signal, hey, come over here, that we get when we have lactose present and glucose absent. Remember, if glucose is absent, that's when we get our cap cyclic AMP flag waving, right? And so when this is a situation, we'll have high operon activity in order to break this molecule apart so we can access the glucose needed for energy. The Lac Operon integrates multiple environmental signals, again, for the goal of saving energy and making the cell more efficient. The next video will be all about how you're gonna see this whole system on the AP exam. So what kinds of multiple choice questions will I see? What will the diagrams look like? What possible FRQs could they ask? This is what we're gonna be looking at in the next video. So please stick around. If this video is helpful, please give it a like, subscribe to my channel for more videos on AP Bio exam skills and this whole gene expression series. Leave a comment, a question, or an emoji down below. And if you're a teacher, check out my Teachers Pay Teachers store, Know and Grow with Gnomes. Keep knowing, keep learning, keep growing, and we'll see you next time.